Hello and welcome. This is an AQA GCSE science video for the topic of infection and response. This is for GCSE combined science biology. Timestamps in the description below. But other than that, let's get going. So let's start off with communicable diseases. Pathogens are microorganisms that cause infectious disease. Communicable diseases are infectious, so they can be spread between animals and plants. Pathogens can be bacteria, viruses, fungi, or something called protists. Bacterial pathogens, there is a bacterial pathogen there. These may reproduce quickly in the body, and these bacteria may produce toxins that damage tissue. This was worth a couple of marks in an exam on a past paper, these 2 points. Viral pathogens, they work slightly differently. They infect and reproduce themselves inside body cells. There's our virus, enters a body cell, and it can reproduce itself. Viruses cause damage to body cells. Next, we have human defense systems. These are non-specific. These are defense systems against all microorganisms. Here's a person. The first defense is in the nose. There is hair and mucus inside the nose or the lining of the nose that can trap microbes. We have the stomach, and the stomach contains acid, hydrochloric acid in fact, and that can kill any microbes that may be present on food. We have the skin, and this is a physical barrier against all microbes and will stop all microbes unless there is some sort of damage to it. If we look inside the body, here are the lungs. We have the trachea, which is the windpipe, and smaller tubules leading off the trachea called bronchi. And if we were to zoom in and have a look, we can see that as the air comes in, the cilia, which are tiny hairs, and some mucus can trap microbes, and the cilia can move the microbes back up and out of the body. If pathogens do get inside the body, we have white blood cells that can help defend against them. The first way they do this is by phagocytosis. Here is a pathogen. Here is our white blood cell. And what can happen is that the white blood cell can move towards the pathogen, engulf or ingest it, and then digest it. So therefore it is destroyed. White blood cells can engulf or ingest and digest pathogens, so they are destroyed. Another way in which white blood cells defend against pathogens is by producing antibodies. Here again are 2 pathogens, and the white blood cell produces antibodies, so there are our antibodies. If we zoom in into that area a bit more so we can see more clearly what's going on, here are our 2 pathogens, here are the antibodies, and the antibodies can attach to areas on the pathogens called antigens, and they can help to destroy and kill the pathogen. So the antibodies attach to antigens on the microbe surface, and they help to kill the pathogen. Another way in which white blood cells can defend against pathogens is by the production of antitoxins. Here's our bacterial pathogen again. It can produce toxins, as we mentioned previously in this slide. There's our toxins. The white blood cells produce antitoxins to counteract the toxins. There are our antitoxins, and these can counteract our toxins. So that's a third way in which white blood cells can defend against disease. Next, we take a look at vaccination. Here is a vaccine. Here is a person taking the vaccine. And inside the vaccine there, we have dead or weakened pathogens. Sometimes you can say dead or weakened or inactive pathogens. There they are. And the way vaccination works is a dead or weakened or inactive pathogen is injected into the body. White blood cells are stimulated to produce antibodies against the pathogen. Memory cells can then stay in the body. On reinfection or on infection with the real pathogen, antibodies are made quickly and in very large numbers. The pathogen is quickly killed and or destroyed. This is worth 3 or 4 marks on a question paper. I've seen it come up a couple of times, so these points would definitely get you the marks. Next, we're going to take a look at 7 types of pathogen and the disease they cause, how the disease is spread, the symptoms, and possible treatment and prevention. Quite a lot to go through here, so let's get going with this one. The first pathogen disease is caused by a bacterium. Bacterium is singular for bacteria. And the disease is salmonella food poisoning. This is spread by eating infected food. The symptoms include fever, abdominal cramps, vomiting, diarrhea, and this is caused by toxins from the bacteria. These toxins are a common theme when we talk about bacterial diseases. The prevention is by vaccinating poultry. Poultry is things like chicken, which is a common source of infection. However, if someone has a salmonella food poisoning, washing hands after using the toilet, disinfecting work surfaces, and washing hands before or when preparing food is prevention. The next one is gonorrhea, caused by a bacterium. This is a sexually transmitted disease. It's spread by sexual contact. The symptoms include a thick yellow or green discharge from the vagina or penis and pain on urinating. It can be treated with antibiotics, and the prevention is something like the use of barrier methods like condoms. The next disease is a viral disease, one we need to know about. This is called measles. It is spread by inhaling droplets from sneezes and or coughs. The symptoms include fever and red skin rash. Fever and red skin rashes. It can be fatal if complications arise. Prevention is vaccinating young children. Another one is a viral disease as well. This is HIV. This is spread by sexual contact or exchange of body fluids, for example, drug users sharing needles. The symptoms start with a flu-like illness, and later on the virus can attack the body's immune system or the body's immune cells, white blood cells, and this can progress to a condition called AIDS, where the body can't fight infections because white blood cells have been or are being destroyed. The treatment, well, this can be controlled by antiretroviral drugs. It won't cure it necessarily, but it can be controlled. And prevention is, for example, the use of condoms during sexual contact and not sharing needles. Another viral disease, this is called tobacco mosaic virus. This infects plants. It is spread by contact between plants or plant leaves. We get a mosaic pattern of discoloration on the leaves, like a kind of yellowy color. This leads to less photosynthesis, less sugars being produced, and less growth of the plant. To treat this, we would remove and destroy, preferably by burning, the infected plants. The next disease is a fungal disease. This is called rose black spot. It is spread by wind or water, and its symptoms include purple or black spots on leaves, which turn yellow, so the leaves turn yellow and drop off early. This means less photosynthesis, less sugars produced, and less growth. Treatment is by removing and destroying the infected plants, or the use of something called a fungicide, which is a chemical that can kill fungi. The next one is a disease spread by a protist. This is malaria. This is spread by mosquitoes, which act as vectors, so they carry the protist and move it from person to person. The symptoms include recurring fever, and in extreme circumstances, this can be fatal. The prevention is preventing the breeding of mosquitoes or the use of something like mosquito nets when sleeping. So there are 2 bacterial pathogen diseases, 3 viral ones, 1 fungus one, and 1 protist one that you need to know and remember. Next, we're going to look at antibiotics and painkillers. Antibiotics help cure bacterial diseases. They kill bacterial pathogens. Specific antibiotics kill specific bacterial pathogens. Remember, antibiotics work on bacteria, not on viruses. So here are some bacteria. Those circles represent the antibiotic, and the antibiotic can kill the bacteria. So there's one type of antibiotic. There is a second type of pathogen in the orangey yellow color, and a second type of antibiotic that's used to treat that one. Bacteria can become resistant to antibiotics. This is a great worry for scientists because if we run out of antibiotics, we will not be able to treat very simple infections. Antibiotics cannot kill viral pathogens. Very important not to give antibiotics to someone who has a disease caused by a virus. The reason why, as we said before, viruses infect and reproduce themselves inside body cells, so it's very difficult to develop drugs that kill viruses without damaging cells and or tissues. Painkillers and other medicines treat symptoms of an illness, for example, headaches, fever, coughs, and that sort of thing. Painkillers do not kill pathogens, they only treat the symptoms. Antibiotics and painkillers and other drugs often come from chemicals found in plants. Discovery and development of new drugs. Traditionally, drugs are extracted from plants and microbes. One example is this one here. This is a type of flower called foxgloves. These were used or are used to produce a drug called digitalis. This is a heart drug and it comes from foxgloves. Another one is the painkiller aspirin, which comes from willow trees. You may have seen willow trees, they're quite common. This one here is a mold growing on an orange in this example, and the drug is penicillin, an antibiotic, and it comes from penicillium mold. This was discovered by somebody called Alexander Fleming. Testing and trialing new drugs. These are tested, the drugs are tested for toxicity, efficacy, and dose. So the steps are as follows. Firstly, possible drugs are selected to treat a targeted disease. Chemicals are either made by chemists or extracted from plants. These go to what's called preclinical trials. These are tested on cells or tissues or animals in the lab. If they are found to be safe, i.e. not toxic, the drug can then go to clinical trials. So we have tested for toxicity. The next step is clinical trials. These are done on healthy volunteers or volunteer patients to test for side effects. Very low doses of the drug are given. Then there are trials done for optimum dose. Optimum meaning the right dose, not too high, not too low. If found to be safe, we go on to trials for efficacy. This means if and or how well the drug works. We do double-blind trials. Volunteers are given treatment with the drug. However, some volunteers are given a placebo. This is a treatment that is identical but does not contain the drug. So here are our volunteer patients. Nobody knows, not the scientist nor the patient knows who has taken the drug and who hasn't. The drugs are then assessed for efficacy, i.e. to see if they work. So here are our volunteers again, and we can see it's worked with some and not with others. And then we test against, we reveal who has taken the drug and who has taken a placebo. And as you can see here, the ticks are the people who took the drug and it seems to have worked on them. Next step is peer review. This is where fellow scientists examine the results, and this is very important because we want to prevent false claims and we want to prevent bias. If you are doing combined science biology, you can stop here. Thank you for watching, and I'll see you soon.